Hello there, my name is Elizabeth McDaniel. In this video presentation, I'm going to discuss the varying manifestations of disease caused by a bacterium called Vibrio vulnificus. This is for Biology 525 Medical Microbiology with Dr. Barrera at Grand Canyon University. So this disease first entered my radar when I moved to the Gulf Coast of Texas about 20 years ago. And living near warm coastal waters means there are always some hazards, and it's important to take precautions when swimming or surfing or fishing. After moving here, I noticed about every 6 months or so there would be a story in the news of a fisherman or a tourist getting struck with this dreaded flesh-eating bacteria. And there have also been several incidences of severe illness due to consumption of raw shellfish. Now, this is not actually a flesh-eating bacteria, but it does cause necrosis of the skin and surrounding tissue. And I will talk about that later on. But this organism, Vibrio vulnificus, is sort of a household term in the lower Texas Gulf Coast and into the Galveston area because it is common enough that people know the term Vibrio by name. So today I'm going to discuss when and how this bacterial disease was discovered, its taxonomy in relationship to other bacterial species, its morphology and metabolic characteristics, as well as its pathogenesis, its mechanism of action, epidemiology, and most importantly, how to avoid infection with prevention tips and what to do if you suspect that you're infected. So I do want to mention also that this can be a pretty brutal disease, and while I try to keep the contents of this video on the tamer side, there are not real images of the actual infection. It does include some graphic descriptions and drawings, so if you are at all squeamish, you may want to proceed with caution. This species wasn't identified as a novel bacterium until 1975, when D.G. Hollis and his colleagues first presumed it to be a variant of Vibrio parahemolyticus or alginolyticus. But Hollis later realized that he had encountered a new species due to its ability to ferment lactose and its lower tolerance for sodium chloride. Later in 1979, this was made official by the Centers for Disease Control. While Vibrio vulnificus was specifically identified in 1975, like all bacteria, it is believed to be much, much older. In the book The Principles of Surgery, written by the Scottish-born surgeon named John Bell, a description of an external puncture wound very closely resembles external Vibrio infection symptoms. Robert Hassing has included John Bell's description in his hypothesis that this was one of the earliest recorded Vibrio vulnificus infections. In Bell's book, he writes about a young fisherman who at first seemed to be recovering well from a superficial wound he acquired on the job. But then a few days later, he developed an extremely high fever, could not keep down any food, and that his wound grew from smaller than the size of a palm to the entire surface of his outer thigh. Bell also explained that the tissue was completely necrotic, stating that the whole skin of the thigh was completely destroyed. The muscles were stripped of skin and fascia from the hip all the way down to the knee. The trochanter was almost bare, the hamstring muscles were exposed, and all of the muscles of the thigh were defected. I have included Bell's image here on the right, and at first glance, it appears to be an early anatomic depiction. So it's almost hard to believe that this was the scaled image of this patient's injury. Hassing also mentioned some of Hippocrates' writing, including similar symptoms after a skin abrasion or puncture wound. And unfortunately, in those days, almost all of the patients died within a week. And if they managed to survive longer than that, their necrosis would progress until they bled to death shortly after. I have already mentioned that Vibrio vulnificus is a bacterium. It is part of the Pseudomonad phylum, which is the most populated bacterial phylum, and is a member of the Gamma Proteobacteria class in the Vibrionales order and the Vibrionaceae family. While there are over 100 species within the Vibrio genus, thankfully only a dozen of those actually are harmful to humans. Among those, and I have them listed here, are three of the most common, which are Vibrio cholerae, which as you might have guessed is responsible for cholera in humans, Vibrio parahemolyticus, which causes gastroenteritis if consumed, and of course Vibrio vulnificus. In a moment, I will discuss the pathogenesis of this bacterium. There are 3 known biotypes of Vibrio vulnificus. Biotype 1 is most prevalent and is the cause of most cases of primary septicemia due to ingestion, but is also seen in externally acquired infections. Biotype 2 primarily causes illness in farmed eels, but has also been isolated from other fish, such as tilapia, and has not been found to cause human illness. And biotype 3, which is relatively newly discovered, is mainly found to cause external infections and has been found primarily in the Middle East. This biotype is believed to be the product of the first 2. Because the second biotype has not been found to cause significant illness to humans, my focus going forward will be on the pathogenesis and epidemiology of biotypes 1 and 3. Like many other highly pathogenic bacteria, this one is also gram-negative. This is noteworthy because its specific polysaccharide capsule evades the body's innate immune defenses, allowing it to take hold pretty quickly. It is a free-living motile bacterium found mainly in marine waters, and it is a facultative anaerobe, meaning it can survive in either low oxygen or high oxygen environments. Vibrio vulnificus will enter a dormant state at lower oxygen levels, appearing to be coccyx shape under a microscope, and in environments rich in oxygen, it will assume its characteristic curved rod shape. This bacterium is also halophilic, which means it prefers salt water. Specifically, it is most prolific in salinities between 20 to 25 parts per thousand. Its motility is due to the presence of a single flagellum, which allows it to travel in open marine waters. Vibrio vulnificus also has unique capsular polysaccharide exterior, which, coupled with several virulence-coating plasmids, is responsible for its extreme pathogenicity in humans. So, how exactly does an infection take hold in the body? Well, there are a couple of ways a person can become infected with this bacterium. The first is ingestion, which can occur through consumption of raw or undercooked shellfish. This is why there's often a warning when you order oysters on the half shell. Ingestion may also occur from swimming in infested marine waters and accidentally swallowing some water. Remember that this is a free-living motile bacterium. Once ingested, depending on the biotype of vulnificus, a person may develop gastroenteritis, which includes fever, vomiting, and diarrhea, which leads to extreme dehydration, or they may develop a systemic infection called septicemia, in which the blood system and body tissues become infected. Or they could develop both. The other infection pathway is through an open wound, which is exposed to this pathogen. Since this bacterium often accumulates in the guts of oysters, this is a common cause for concern in commercial oyster shuckers. Puncture wounds from fishing hooks or shells on the ocean floor may also harbor this harmful bacterium. There are 4 major exotoxins which are responsible for its extreme virulence: cytolysin or hemolysin, the Martex toxin, secreted phospholipases, and vibriolysins. The first, cytolysin, also called hemolysin, works by interacting with cell membranes to form a pore made up of beta barrels. That's this one right here, through which it is able to enter erythrocytes and essentially steal the iron from the hemoglobin. Once this occurs, cells will self-destruct through apoptosis. So this explains the flesh-eating misunderstanding I discussed earlier. Vibrio vulnificus is not technically consuming flesh, but it does sort of usurp our necessary proteins and iron and lead to the destruction of our cells. Another exotoxin present in this species is the Martex toxin. Martex stands for multifunctional auto-processing repeats in toxin, and this one works in a similar manner. It binds to the cell membrane and creates a pore which opens into the cytosol, only this time it doesn't directly target red blood cells, but instead effectively throws a wrench into the machinery of the cell's organelles. So this can be in other tissues as well. Or I should say it throws multiple wrenches in the cell's machinery in the form of effector domains, which target multiple areas within the cell. It inhibits autophagy, disrupts the Golgi apparatus, and ultimately leads to lysis of the cell. And as if that wasn't bad enough, there are two more. The third major exotoxin is a series of phospholipases. As their name implies, these compounds effectively demolish the structural integrity of the cell's lipid membrane. And of course, a cell is no longer a cell without that phospholipid bilayer. And finally are the Vibrio genes. These are present in all other pathogenic Vibrio species as well, and there is still some ongoing research in this area. But what is known is that there are several genes involved in upregulating the chemical factors that are secreted in Vibrio vulnificus and other species. The Vibrio encompass the toxins that target extracellular components, such as the blood vessels and extracellular matrix tissue. Essentially, these exotoxins target cells by invading the cell membrane, that's these three, and releasing harmful substances into the cytosol. They also destroy the plasma membrane directly, such that's this one again, and they harm bodily tissues directly in the extracellular space. The good news is that this horrific disease is comparatively rare. In the United States, there are fewer than 100 cases per year. Interestingly, more than 85% of people affected are male. This may be due to the higher prevalence of men holding jobs in the fishing and oyster industries. In addition, people who are immunocompromised due to HIV infection, organ transplant, or inherited immune deficiency are more likely to become ill after an encounter, as are people with liver diseases such as cirrhosis. As you can see from this map here, most of the infections within the United States occur in the warm waters along the Gulf of Mexico, between Texas, where I live, and Florida, with a few more extending up into the Virginia and New England coastline. Additionally, infections peak in the hottest months of the year, which for the Atlantic water temperatures means July, August, and September, with water temperatures ranging from the upper 70s to mid-80s. Likewise, the lowest infection rates, understandably, occur in the cooler months of December, January, and February, when colder water temperatures send free-living vulnificus into a dormant state. The images shown here reflect trends in the United States, but similar patterns can be seen globally. Warm subtropical waters yield higher rates of infection, especially in hotter months, and cooler latitudes with temperatures averaging below 10 degrees Celsius yield virtually no infection rates from marine waters. There are recorded infections in Southeast Asia, along the Israeli coast, and in South America as well. The exceptions to these are in areas which import shellfish from these warmer waters. And if you are wondering about how climate change has affected infection rates, you are in good company. Archer and his colleagues have investigated this very thing and have found an average annual increase from the 1980s, when the yearly rate was around 20 occurrences per year. After all of that, if you're like me, you may be curious about how you can avoid this ill-fated infection. Well, prevention is actually pretty straightforward. Avoid swimming with an open wound. A bleeding cut or puncture wound is an easy target, not only for this pathogen, but other marine bacteria as well. Second, do not eat raw shellfish. The CDC recommends boiling oysters for 3 to 5 minutes after the shells have opened and frying for at least 10 minutes. The internal temperature of fish should reach 145 degrees Fahrenheit as well. Safe food handling practices also apply here. Make sure to thoroughly wash your hands if you handle or prepare shellfish, and clean any cuts acquired from shucking oysters thoroughly. If you are swimming and happen to step on a sharp shell or piece of glass, make sure to wash it thoroughly with soap and water and apply an antiseptic to the injury as soon as possible. And most importantly, if you notice any symptoms of illness after doing any of these first 3 things, you should seek medical treatment as soon as you can, and make sure to mention to your care provider that you may have been exposed to this pathogen. Once infected, treatment includes 1 or more strong antibiotics, debridement of the infection site, and in some cases, amputation. Fortunately, medical treatment and understanding of this pathogen has improved greatly in recent years, so infection is no longer an immediate death sentence. That's all I have for now. Thank you for suffering through this video, and if you'd like to know more about Vibrio vulnificus, I have included my references on this slide and the next two.